So Akis is what I call a breath of the Mediterranean in a German country. He started in Greece and then moved around the world, UK, and finally set shop at the end of the first decade of the 2000 in Germany. And now he's in Translational Epigenetics group in the University Medical Center at Göttingen. Now, he described his research as trying to figure it out or helping to understand the rules that regulate gene expression in response to regulatory elements inside the nucleus, then he says explicitly that the structure of the nucleus or the architecture he used is very important. And in fact, I followed him for the last years, and this is what he does. Actually, he's not lying to us. He's trying to tell us what the heck is going on in those nucleus. In parallel to that, he's been very much involved in what you will see next, which is this DFG large-scale project, SPP 2202, whatever that means. And he, together with a few others, have been coordinating this effort, and I guess we will hear about this a bit more. So Aki is super happy to have you with us as well.
So I should start by saying that I'm really happy to be here. It's a great idea by Marc and by our INC consortium, and I'm really happy to represent both the SPP 2202, which you see here, and the INC in a way. So SPP stands for Schwerpunktprogramm, which is loosely translated into a priority program. And these are 3+3 year programs funded by the German Research Council to promote a sort of cutting edge field that the community and the DFG feel are important for Germany, and to promote them, expand them, and create a tightly knit community. So a couple of years ago, we tried to initiate such a program, and the idea, of course, was that there was, enough expertise within Germany of people that study 3D nuclear architecture in different ways, and also at the same time that there was enough evidence in the current literature that this has been a very important field of study that keeps giving us really cutting edge new information about how the genome is structured, regulated, and deregulated in disease. And as you can see, what we put together in the end was this idea of spatial genome architecture and how it's involved in development and disease. The people behind this, so there's lots of group leaders, which I'm going to present, but the people that mostly initiated this, always in discussion with the broader community in Germany, were Stefan Mundlos from the Max Planck and Charité in Berlin, myself, Anna Pombo from the Max Delbrück Center in Berlin, who is also part of 4DN, and Eileen Furlong at the EMBL Genome Biology Unit. And we are the people that are sort of dealing with most of the coordination, but it's a consortium where everyone is heavily involved in most of its aspects. So what was the mission? So, this being Germany, we needed a pretty defined mission for this consortium, at least for the first years, and here it is. So we wanted to exploit this still emerging field, and this is exactly the text that we provided as guideline for the applications. I should say here that it's an interesting call, right? Because you design it and you propose it to the government as on the basis of some idea, and you propose people that would be. But then what the DFG does is it makes a completely open call to the whole of Germany. And then there's a completely autonomous review process that we have nothing to do with. So we end up getting the best project according to outside reviewers, both with our on-site visit and people looking at individual grants. So the idea was to bring together a fairly large and interdisciplinary group of scientists that can consolidate this topic, which was already present in Germany, but also advance it within this 3+3 years on the basis of collaboration that are going to be built, but also with individual projects applied for. And then the idea would be to apply novel and existing technologies and also further develop them in order to address, of course, the long-standing question that most of us has, which is what is this codependency, this relationship between the structure of the genome and its function. The aspect here, I think this is important to stress, because we were in the same greater realm of projects like the 4D nucleome. We wanted to have elements that sort of made us slightly different and did not overlap, so there wouldn't be redundancies on the broader picture of 3D genome organization and the field. So we wanted, almost by definition, a more functional and mechanistic studies using either in vitro or in vivo models that have been challenged in some manner. So either during stimulation, disease, differentiation, early development, carcinogenesis. And then the idea was to understand the mechanisms behind it, and especially in the context of disease, to follow up on all the pioneering work, for example, from Stefan Mundlos' lab on how disease might actually manifest via changes in 3D chromatin folding. And the last aim that we described had to do with novel computational tools and models that would allow us to either predict folding dynamics or explain them more easily and then translate them into some sort of genomic output that is measurable. And this is, an example of this is what I'm going to talk about. And actually, by sheer chance, it fits very well with what Jan was just describing just before. But we will get. So I'm going to very briefly present the people that are part of this. Many of these names, I'm guessing you will realize, they are PIs from all different places in Germany that have very diverse interests as regards the systems that they study in and the way they approach them. And I have an overview slide in a bit. And these people, again, have been selected independently of us by our peers outside Germany. And they've been brought together in different teams with different projects. And the nice thing is that we can support 24 PIs in a large number of projects that have, a couple of PhD students each. So it is a generally large consortium that is also training a decently large number of people at the same time. And here you can see the 20 people that are currently part of this consortium, together with the 4 people from the steering committee. And of course, you recognize some of them, but if we try and sort of artificially group these, because of course, many people, including my lab, we don't just do one thing, we try and reach out to other parts of this. We have a number of people that focus on C-type methods. Anna might object a bit because, of course, GAM is not necessarily based on 3C, but, broadly speaking. So they use molecular methods to identify changes in 3D genome organization and try and stratify them and understand them. We have a very strong core of people that have been, long interested in imaging, either developing new imaging techniques or applying them broadly to look at changes in genome organization. And this, ranges from super resolution imaging that the Leonhard lab does, or the Cardoso lab do in Munich and Darmstadt, to high throughput imaging that Vasilis Zoukos does at the IMB. And then, of course, we have a number of people, and I've sort of deliberately put myself here because I'm going to talk about bioinformatics method, that are either involved in the computational analysis of these, the computational modeling, or the development of new algorithms and tools to be able to work with this sort of data. However, what again is interesting about this consortium and sort of discriminates us from, well, to some extent at least, we think, is that we are actively, each project has a mechanistic goal to address, a fundamental or hopefully fundamental mechanism that connects gene expression with 3D genome regulation. And what we do have as well is a very diverse number of model organisms that we work with. So there are groups that, of course, have in vitro models ranging from normal cell lines to iPS-derived cells and organoids, all the way up to zebrafish and transgenic mouse models, to fruit flies, Hydra, surprisingly perhaps, and of course, man. And all these, and of course, when I talk about man, we also have patient samples, for example, directly patient samples and cancer samples. So all these sort of create a heterogeneous but very interesting mixture that meets very often. So we have an internal sort of lab meeting once per month, and we are really, to my surprise, in some sense, a very tightly knit community that discusses projects and tackles problems and gives feedback to one another, and of course exchange techniques, students, approaches and models. So we are, our consortium is coming very close to the end of its first funding period, and we will be applying for the extension for the next 3 years. And of course, the faces in the consortium might change to some extent as people move from country to country, or as projects renew themselves or not. Again, this is a completely open call, but it is remarkable that the German government gives you this tool that has not got the magnitude of the 4DN funding, or let's say of previous large projects like the ENCODE, but it creates a starting point for the German community to really move forward. And of course, within it, as I said before, we have Anna, for example, who is also part of the 4DN, myself, who is part of the IN consortium together with Anna, and people that are involved in different sorts of international or transnational consortia of this sort that help with the outreach of our SPP project. So I'm happy to address any question that you might have about this, but I was advised that this is also a science talk, and I thought that it would be interesting to talk about the actual projects that we have running within the bounds of this SPP. And this is the brainchild of our bioinformatician, Eduardo, whom you see here, being very pensive. And he uses data from Sia and Lily to do what this he's doing. So I'm going to delve right into this, and I'm actually going to spell out the whole truth of how this came about. And again, I'm thankful to Janma for sort of semi-introducing this. So back in 2016, we had come up with an idea of capturing the native conformation of chromosomes. And the idea was that we would come up with a protocol that would allow us to do high C or 3C-like experiments without the need to cross-link. And so this is all published. I'm not going to go into details, but of course, the problem here was, although most of it worked very well, that when you looked at a genome-wide picture, which you can clearly see here, if you compare a normal in situ high C to our I high C, or intrinsic high C as we call it, it is pretty clear that one looks like a beautifully structured chromosome with defined compartments, and the other one looks like a compendium of noise that tends to structure in some way, but doesn't really. So the problem has always been, ever since this first paper, how do you analyze such sparse data? Because, for example, finding these local maxima that represent the CDCF loops that we know very well about is very difficult here, because every little dot essentially represents a local maximum, more or less. So I presented this problem to Eduardo a few years back, and he started heavily thinking about it. And he came up with a process called Bloom. And I'll try and explain this to the best of my ability. So here you see what the normal data look like, the crosslink data. This is data that can afford, let's say, 5 or 10 KB resolution. So when we plot them at 1 KB, they sort of break up. They're still structured, but it's sparse. This is our unprocessed native data. You can see it's very sparse, and the more you zoom in, the sparser it gets. There is actual signal in this last 1 KB resolution box, but it's sparse and it's weak, so it cannot be easily visualized. But when Eduardo magically applies Bloom to this data, we get a picture that I would argue is very close to the data we understand from the crosslink data, but also very rich in interaction all the way up to a 1 KB resolution map. And of course, this is only visual, but we've done, and I'm not going to show you everything, but we've done a number of tests looking at a number of features that we would expect to sort of be enhanced and show up in these maps. And actually, we can show that we have very, very strong detection of a number of features that we were expecting, an interaction we were expecting to have these maps. And in many cases, these actually surpass the resolution that you can get from in situ high C, even enhanced in situ high C, or even more highly sequenced. You can see, for example, the polycomb to polycomb interactions that become really sharp and defined, or those between poison enhancers and their target genes. Now, of course, the question is, how does the magic happen? And I'm going to, again, to the best of my ability, not going into the maths behind it, to try and explain. So if this is the sort of noisy initial matrix that you're starting with, the whole idea is the following. I typically present this as the inverse of deconvolution that people do when they do microscopy. So when you have a picture that has lots of overlapping signal, you do deconvolution, trying to find the local maxima of the different signals in your picture and make them sharper so you can discriminate two objects from one another. You can think of this as the inverse, that you have different very focal points of signal in your high C matrix, like the one you have here. And then you're asking whether the individual little signals actually belong to a different but specific distribution of signal that is larger and engulfs them. And this is not something that we magically came up with. This is a known principle in mathematics called the Dirichlet process, which is fitting data, distribution of data, with the best distribution that they represent. And the way to do this is to start with your own matrix and first do a balancing of the matrix like you would. We do not use the typical, for example, KR or IC balancing, although they would work too. We use something that's called an Osborne balancing method. Again, not something we came up with, something that is known in mathematics. And then we run the Dirichlet process across our high C maps on a per chromosome basis in different directions across the map for a defined amount of time. And here you see examples of how these maps are gradually transformed by our algorithm finding and fitting signal distributions onto the presumed noise that you have in this matrix. So you go in one direction, in another direction, so you move it in different ways in the matrix, and then in the end you end up having this Bloom map, which at least visually to a layman biologist like me looks like a convincing high C map. Now, again, I'm skipping much of the bits and pieces that have to do with validating that this is indeed a compartment, and you find the same compartment, you look at crosslink data and it's the same position and it has strong or even stronger insulation and so on and so forth. What can this be useful of? Because we never intended to use this tool as an imputation tool for mostly, but we actually found out that it does something very spectacular that I'm going to come to in a moment. So we've also applied this to single cell high C data. So this is deep C data from the Sunny Z lab in lymphoblasts. And here you see the three individual cells that we're giving as an example, and how their maps are transformed in something that is essentially an imputation, not the same way that Jan just talked about, but again using the Dirichlet process. But the very interesting thing here is that we can really get KB resolution maps where you can actually find individual enhancer promoter interactions that are cell specific, so they belong to individual single cells. And here we have the example of the EZH1 gene and how you can realize some of this signal even if it's not as strongly there in the initial data, and so on and so forth. And you can get this signal, but not that one, because presumably this interaction doesn't exist in these cells. And then the original DeepC paper used some of the contacts, the cell type specific contacts in the data set to actually look at their diversity. And this is the plot that they generated. But after we bloom data, I think you can appreciate that we have much more accuracy in bringing these together just by looking at interactions that are, between enhancers and genes specific to these cellular subtypes. So this, we think, gives us a tool that we can do this sort of imputation step and also look at, really high resolution contacts in this sort of data. I should say that we've only tried it in this deep-sea data, which to my knowledge is one of the best data sets out there. It's, it's dense for a single cell data set, so there is stuff to work with. However, the interesting part of this came when we did a test. So we took the 1 megabase domain that encompasses the CMYC gene in normal human endothelial cells. And we use this domain because the MYC promoter and its enhancer contacts are very well characterized. So we use different tools in normal, either uncross-linked, so this is our I-Hi-C, or crossing data to process this data and call enhancer promoter interactions. So this is data from the Rao paper, so it's pretty high resolution, very well sequenced, and this is our data from HUVEX again, before and after bloom, so processes after bloom. And I think you can appreciate the contact interactions that we get after processing the raw in situ Hi-C and the ones that we get after processing our data. However, we went on to actually test whether the interactions that we map are actually true and whether they have an effect. So we have these 5 very well defined, also functionally defined, enhancers of the promoter of MYC that you see sitting here. And this subtab is 0.5 megabase in size. And then what we did is we used CRISPR-I to interfere with each one of these sequentially in triplicates. And what we found is that, of course, interfering with each one of these 5 enhancers gives you a different extent of downregulation of the gene. However, the change in expression level correlated almost, well, anti-correlated almost perfectly with the interaction strength, which is a measure that comes out of the bloom processing. So there's a number behind this bin that connects enhancer 1 to the promoter of MYC. And that number is, in this setting, absolutely predictive of the extent by which this enhancer controls the expression of the MYC gene. And you can see that enhancer 1 is actually the strongest effector of this gene, although it's not the closest one. And enhancer 4 is the 4th strongest, and the 2nd strongest is here in the middle, and so on and so forth. This was very surprising for 2 reasons. The first reason is that bloom was not necessarily designed to do this, so we didn't expect that it would actually return this result. And the second reason was that this implies that there is information in the Hi-C data that can be somehow extracted that tells you whether a given enhancer has a strong influence on the expression of a given gene or not. The problem, of course, is that this only comes from one locus, that it has a very specific structure, harbors essentially just one gene and its regulatory element. So it's not representative of different regulatory scenario in the genome. So the closest we could get to that was to use the Gasparini CRISPR enhancer screen, which is a beautiful paper from 2019 from Jason Durer's lab. Here they've screened almost 6,000 enhancers with multiple guide RNAs in an unbiased screen in K562 cells, for which we also have, very good quality, high resolution, Hi-C data. And they ended up identifying 664 very robust enhancer gene pairs that they could claim that they could knock out strong enough to see measurable differences in the promoter that they talked. So what we did is we took K562 cell Hi-C and we bloomed it, and then we used, and we actually bloomed it at a sub-KB resolution, because you can ask bloom to sort of modulate the resolution of the resulting matrix. And then we used the bloom score at the interaction of individual enhancers with the target gene as defined by the Sindura screen to see how well we can predict what they had already experimentally validated. I've chosen to show you the RNF example here because this is a very small region of the genome. This is actually less than 20 kb. And I think you can appreciate that despite how funny looking the Bloom Hi-C map might look, we can actually decipher three very specific interactions between three defined enhancers from the Gasparini screen. So to cut a long story short, we actually have a spectacular correlation between what Bloom predicts as a strong or weak enhancer with the change in expression of the gene upon knocking out of that enhancer. So the R-squared value is 0.94, which is essentially too good to believe. If one uses any other of the sort of state-of-the-art tools, and now we've added more to this collection, you really cannot recapitulate this. Although, you know, some of them, Hi-C, for example, doesn't perform that bad given high resolution data. But the main reason why Bloom performs so well is because it has this precision in the score that you get. So with every other tool, you get some sort of an interaction score that pretty broadly can correlate with many different expression quantiles. Whereas in our case, it's a pretty defined scale throughout the whole scale of expression changes. And just to add to that, this amazing correlation seems to be completely independent of individual features that are present at these enhancers. So you cannot explain it by simply the strength of accessibility or histone mark levels or the presence and strength of nearby CTCF. And you also don't seem to be able to at all explain it with models that are rather popular nowadays, like the ABC model. So given that, we are now becoming more and more confident. I should say we've analyzed another screen, which is smaller scale, but actually analyzes multiple enhancers against one given gene across 7 loci. And there, Bloom also performs with such remarkably high accuracy. So we think that we have sort of inadvertently come up with a tool that allows us to make functional predictions about enhancers and the genes they regulate only on the basis of Hi-C signal, which again, on the one hand seems very surprising, but on the other hand also highlights that there must be something inherent in what Hi-C measures that actually gives you strengths and functional interactions, again, to the extent that we can test, you know, a large number of enhancers. And of course, the promise here, or the hope, is that since we have all these Hi-C data sets out there, and some are, you know, higher resolution than others, but it doesn't matter, you can actually use Bloom to enhance them, take out all these enhancer promoter contacts, and then make predictive functional atlases for any sort of Hi-C data set out there from different tissues or cell lines. And of course, hopefully one would be able to do it from quality single cell Hi-C data, which is an aspiration for later in time. So with that, I've come to the end of my presentation, and I'm happy to take any questions if there are any.
So thank you, Akis. Beautiful. The Bloom seems magic to me.
To me, for a very long time.
Because you really are squeezing a lot of information for something that under the eyes looks totally flat, but obviously it's not flat, otherwise the algorithm would not pick up the signal. So you have shown several validations that you have done, at least in those few slides. But I wonder if you could take something like, I don't know if you've seen this latest bioarchive from Luca Giorgetti.
Oh yeah, the predictions of the enhancer in the
this is experimental. Yes. The title is something non-linear correlation between expression and distance between enhancer promoter. That data set would be beautiful if you could actually predict what they find in the actual screening that they do. Have you thought about blooming that somehow?
So the problem is they don't have high-C data from all of that. It has basically been done on the basis of imaging. So we wouldn't have data to bloom for everything.
Well, you would have the wild type.
We have the wild, yes.
You will have, because I think it's done in one cell line that is
it's done in mouse cells, right? And they have this jumping.
Yeah,
sorry, there's
exactly.
a helicopter flying outside my window, so it will go.
We don't hear it.
Oh, good. You will in a bit. So it won't be possible to use that particular data set. What we are doing, which I think, so I have the same problem like I guess everyone would have. We cannot believe that a technique that has potentially biases and has a subset of things that it's looking at can have functional information embedded in it. And this has been the issue why we kept the preprint from even posting it on Bioarchive for a very long time, because I refused to believe that this data was true. I got convinced when we analyzed the Gasparini data. So what we're trying to do is two things. First of all, we're trying to artificially remove, we've done this actually, we artificially remove signal from high-C maps. And we're giving this high-C map that has a stripe that's completely empty to bloom and say, well, come on, magically put something in there. And it will not. So it doesn't create signal out of nothing. So it needs the sort of surrounding signal to be able to fit distributions and try and infer what might be coming there. But this has only to do with the presentation bit of it, because the problem, of course, comes with the accuracy of these values that are being assigned to the individual interactions. So the way to address this would be, as one of our reviews has very cleverly suggested, you can take a 3D model where you know the precise number of contacts between different beads in that model. Do 1,000 iterations, use half, bloom them, and then see whether you're recapitulating what you get in the model. That has proved more challenging than what we thought, but we are getting there, and it seems like it works. So it doesn't seem to work with every type of contact. So again, brings me back to this idea that there is something inherent in the biological noise that exists in the high-C data that allows you to do this prediction. And the same applies to when you either add artificially noise or remove signal from these maps. There's a finite number of noise that you can add before bloom doesn't predict well anymore, and a finite number of noise that you can, a signal that you can subtract before bloom starts having problems, which is a reassurance that it's not really magic and it doesn't make things out of nothing.
When can we see bloom? Because I went into the GitHub.
Oh, this is a long story. There's many people that have been asking for it, and we had a version on the GitHub which was really ugly and dirty. So I explicitly said that we take it down. Ever since 4 weeks now, we have a clean version with some modifications that is going up in the next week. So you'll be able to see it. So it will be there. We're finally, very close to also finishing the revision. So
there is one additional question from Emmanuel. Thank you for the talk, super interesting. Have you tried to compare with micro-C results with the high-C data sets analyzed with Bloom?
Yes. Our sort of KB, sub-KB resolution maps are not necessarily micro-C maps. They don't necessarily look the same. We actually generated a micro-C data set in that direction, and we are doing that. Yeah, this is the best that I can say right now. They don't necessarily see exactly the same things.